Hi, everybody, and welcome back to Miss Angler's biology class. I am Miss Angler. In today's video, we're going to be looking at digestion, and this is a follow-on video from the small and large intestine. We're going to focus in on the processes of chemical and mechanical digestion, and we're going to see what are the structures required for these things to happen. Now, if you are new here, don't forget to give this video a thumbs up and make sure you're subscribed with your notifications turned on because I post every Tuesday and Thursday for grades 10 to 12 biology. And if grade 11 is really getting you down right now because there's just so much to learn, you should think about getting a copy of my cheat sheet study guide. It makes learning and studying so easy. It's literally taking pages and pages and pages of your textbook and putting it into one easy page of study notes. So let's talk about why do we go through digestion. Now, when we look at our mouth, we often don't think that digestion is actually necessarily happening in our mouth, but it is. And there are many important processes that begin the digestive system or the digestion process right here in our mouth. Now, ultimately, the overall goal for digestion is to make smaller particles, okay? So keep that in the back of your head when they ask questions like, why do you chew? Why is there enzymes? Why is there this? Why is there that? The end goal is we want to make smaller particles so we can increase the surface area. Because if you increase the surface area, you can do a lot of digestion a lot quicker and you can access the nutrients a lot faster. Remember, everybody, that when you eat like a hamburger, that carbohydrate of the hamburger bun, that doesn't just like go straight into your bloodstream. Otherwise, you would just be made out of hamburger bun and everything else that you've eaten. Instead, we have to take that very large structure that is insoluble, doesn't dissolve in water, it doesn't move through your capillaries. We've got to make it really teeny tiny so that it can be absorbed into your tissues. Now, this whole process is in two parts. One is mechanical, and as we see here in the mouth, there are a couple of things that we've already covered when it comes to what's in the mouth. We already know that we've got teeth, which are responsible for mechanical digestion. I don't know if you know this, but also the tongue technically counts as a part of the mechanical digestion and moving that around in your mouth. And then sitting in the back of your throat and on the top of your mouth, which is a little bit invisible in this picture here, but you have your salivary glands, and they start the chemical digestion. So let's get into some more specifics about mechanical digestion beyond the mouth. The mouth is where mechanical digestion is going to begin, with your teeth and your tongue. But a lot of people don't realize that mechanical digestion continues going down the esophagus. It technically counts as a part of the mechanical digestion because of the way in which the esophagus moves. Now, what you can see here in this picture over here is you can see a bolus of food making its way down the esophagus. And ultimately, what happens is the esophagus is a tube of muscle which is slowly squeezing the food or the bolus down to the stomach. And I'm sure we are familiar with this term. We use the term peristalsis, which is the contractular wave movement that squeezes the food from the top of the esophagus down the esophagus to the stomach. Now, speaking of the stomach itself, often we don't think about the stomach being mechanical digestion because we think, oh, it's all the juices and the liquids, so it must just be, chemical. But that's actually not true. We still have mechanical digestion taking place inside the stomach here, and we can see a lovely cross-section into the stomach. First of all, let's take note of the fact that the stomach is made out of smooth muscle. And smooth muscle, everyone, remember, is involuntary muscle that can contract and relax. And it's that contracting and relaxing, which we call churning, that is what mixes your food so that it's evenly digested throughout. And that counts as mechanical digestion. Now, we can mechanically digest food up until a certain point, but there's a limitation to this, right? You can chew and chew and chew food and mix it as much as you want, but eventually it will become a liquid, but it's not small enough for any absorption to happen into the bloodstream. And that is where chemical digestion takes place. Now, there are three major places that chemical digestion takes place, starting off with the salivary glands. Now, here is a little diagram over here, which we did cover in my introduction to the digestive system, which was the set of our salivary glands, which is this one over here. And we've actually got three sets of glands in our mouth, and they secrete out saliva, which contains enzymes. Now, this is not the only place that enzymes are secreted, but it is where chemical digestion, specifically for starch, begins. Now, the main attraction for chemical digestion is always going to be the stomach, okay? What we see here in front of us is we have a liquefied version of the food that we have eaten, and that is due to all of the substances that we have added to it, including, most importantly, hydrochloric acid, as well as, of course, those enzymes, because we do also have enzymes in the stomach. Now, I'm going to elaborate on these enzymes a little bit later, but I want you to know that enzymes perform their job best in an environment that suits them, so their pH and their temperature, and that's why we find enzymes in different locations. We have them in our mouth, in our stomach, and our third and final location, which is in the duodenum, which is the first section of the small intestine. Now, I want you to know, everyone, that after the duodenum, no more chemical digestion takes place. Absorption begins. And it's important to know that because ultimately the enzymes don't continue doing their process forever and ever and ever and ever. There is a limitation to how long this goes on for. And so digestion needs to be roughly completed by the time food leaves the duodenum. Now, why are we going through chemical digestion? Ultimately, I want you to think of it like this. When you eat food, you are eating food that is in a complex structure. So we're touching on our grade 10 knowledge, and we're thinking of chemistry of life. When we look at something like, for example, a protein, if I draw it for you here, is made up of amino acids. Each one of these circular structures represents one amino acid. And so if I join enough amino acids together, I form a protein. Now, when you eat whole pieces of protein, like you eat a piece of meat, that piece of meat is not useful to you, even if we liquefied it, so we mechanically digested it. We need to get it to its simplest form, its amino acids. And so to do that, we use enzymes and hydrochloric acid to cut these chemical bonds between the amino acids so that we can get them as individuals, because we want them, remember, the smallest pieces. Remember, that's the goal. And we do this with carbohydrates, with proteins, and with fats, but we do them in different ways and in different locations. And so that brings me on to a really important part about whether or not a substance is soluble or insoluble. Now, when we speak about chemical digestion, we are ultimately taking something that is insoluble in specifically water, and we're making it soluble in water. And so what that means is that before we did any digesting, just like we did earlier when I drew you this diagram down here, this would be considered an insoluble substance. It's too big, it's too complex. We want to make it smaller, so we just have these individual parts which are soluble so that they can be absorbed into the bloodstream. So now we need to talk about the star of the chemical digestion, which is enzymes. Now, I'm not going to go into all of the basics of enzymes in this video. If you can't remember everything from grade 10, then click the link above now, the little video card that will take you to my enzyme video. Go watch that, refresh your mind on what enzymes are and how they work, and then come back here so we can see how enzymes are used in the digestive system specifically. Now, ultimately, when we talk about enzymes in the digestive system, we want to know why we use enzymes. And the first thing we need to talk about is we use enzymes because without them, our metabolic rate would be too slow. And so if you were eating food, it would take so long to digest food that if you rely just on hydrochloric acid to digest your food, it wouldn't take a few hours. It could take days, weeks before food is completely digested with the hydrochloric acid, and that's just not optimum. You would die before then. And so what we also need to remember is how enzymes do their job. And remember, this is just a refresher on these basics we need to know. But remember, enzymes can be catabolic and they can be anabolic. Catabolic means they break down, and anabolic means they can break up. And all enzymes can do both, depending on how much substance you have. If you need more, you'll build up. If you need less, you'll break down, and so forth. So it just depends on what the body needs at the time. Now, another little refresher that is key to the digestive system is this little cutout that we see inside of enzymes. Now, that little cutout is called the active site, and that is where the chemical reactions take place. And I want you to be very aware that the digestive system has certain pHs for their enzymes to work. Now, this is just a summary of some of the pHs that you may need to know. Depending on your school and your teacher, you might need to know even more than what is in this table, but these are the minimum. Now, you will notice if you look at the table that there are specific pHs that certain substances need to be in. So, for example, starch needs to be in a pH of 7. Protein needs to be in roughly a pH of 3, and lipids or fats need to be in a pH of 8. Now, if you know the digestive system, you will know that the pH changes regularly. Your mouth begins at a pH of 7. Your stomach can go all the way down to a pH of 2, and then your small intestine goes back up again to a pH of 8. And so it's really important that the enzymes that exist in the mouth and the stomach and the beginning of the small intestine all have the right pH, because if they don't, what do we know happens? Again, this is a little bit of revision. We know if we have the wrong pH or temperature, we end up denaturing or changing the active site. Remember the basics of bio. If you change the shape, you change the function. And so we got to keep that in mind, because if we want to be able to digest a substance, we have to be able to digest a protein with a protein enzyme. And most of the enzymes associated with the digestive system have a name linked to the substance they are digesting. So, for example, the classic 3 enzymes we generally need to know are amylase, and it's known as a carbohydrase. Enzymes generally end in the word ase or the suffix ase. We have trypsin, which is a type of protease or protein enzyme, and lipases, which are fats. We also, in exams and tests, need to know the origins of these substances, these enzymes. And so first of all, we see amylase coming from your salivary glands and the pancreas. Protein and trypsin enzymes are seen in the stomach, pancreas, and small intestine, whereas lipids are exclusively from the pancreas. Now, I can't stress enough to you the importance of knowing your enzyme characteristics. And the reason for that and how it links into the digestive system is if you understand their characteristics, you will be able to understand their role in the digestive system and how they are affected. This is often where a lot of the application questions come in the exam as well. So we need to remember that enzymes are reusable. Often you get asked, why do you only need a small amount? And that's because they can be used over and over and over again. The other thing that's really important to understand is that enzymes are substrate specific, which means in the digestive system, if I digest fat, I can only digest fat. I cannot digest protein. I cannot digest carbohydrates. So that's why I need to have an environment that allows me to do my job efficiently. Also, what's important, as we just covered, is that they are pH specific, and I'm going to come back to that as we round off this video. And last but not least, as I mentioned just earlier, that we only need a really small amount of enzymes because they're so good at their job that they are reusable and they just do their job over and over and over and over again. And they require small amounts because they do their job and their metabolic functions so quickly. Now, one really important thing that I must clarify and that we must get right before the end of this video is the enzyme action location. This is the thing that we get wrong time and time again in exams. So let's just quickly recap about the pHs of the various enzymes. When we're in the mouth, the pH is roughly a 7, which is fairly neutral. The stomach is an acidic pH of 2, and then the duodenum and the small intestine are both a pH of roughly an 8, which makes them alkaline. Now, this is important because it ultimately affects how the enzyme is going to work and where it is allowed to work. Now, when we're reading this diagram, it's also important to understand, what we're looking at here. Every time you see an arrowhead like this one over here, it means that the digestion of that substance is taking place. If there's no arrowhead, like we can see over here by lipase or lipids, there's no digestion happening. And so we need to keep that in mind as we go through this location of enzyme action. Now, we're going to start off with the carbohydrates. Now, carbohydrates begin their enzyme journey in your mouth, and they interact with salivary amylase, which loves a pH of 7, and they become polysaccharides and disaccharides. You will notice in the mouth as well that there is no digestion of lipids or proteins happening. You'll see there's no arrowhead there. It's just like a solid color. So that means that no digestion is happening in terms of chemical digestion. So the only substance being digested in your mouth is a carbohydrate, okay? And that's because the pH is just right. Now, what happens when we go to the stomach? We go into the stomach and the pH dramatically changes, okay? Dramatically. You will notice now in carbohydrates, nothing is happening in there. And that is because amylase cannot perform its duty in a pH of 2. Neither can the same happen for lipids, and so they don't get digested there either. However, we notice here that proteins do get digested, and they are going to be digested into polypeptides via an enzyme called pepsin. And that's just one of the protein enzymes that I've mentioned. I did also mention trypsin earlier. Now, that means in the stomach, the only substance protein. So those are the two things that are important. And you'll see we're keeping their digestions separate from one another, carbohydrates and proteins. But now we get down to the small intestine, which is where it gets interesting and it gets a little more complicated. The pH of the duodenum is 8. And you will notice that carbohydrates come back into digestion via pancreatic amylase, so not salivary amylase, but pancreatic amylase. And you'll see there's digestion happening here, which forms disaccharides. You will also notice that protein is also happening in terms of its digestion, and it is becoming peptides with the use of the enzyme trypsin, which I've mentioned earlier. Again, you will still notice no digestion of fats are occurring. Fats are really tricky to digest, everybody, and you may or may not remember this, but I did mention in my introduction to the digestive system that in order to digest fats, we need bile. And so bile is really important, and bile only comes into play in the duodenum, which we can see here. It says bile salts and the enzyme lipase. And so we only add them then, right? They only actually get added then. It's once we are in the duodenum with those substances, and this is just the beginning part, remember, of your small intestine. Now that you've added in everything, you are now able to digest protein, as we can see here. You are able to digest carbs. And now finally, you are able to digest fats in this final section. Remember, fats need to be emulsified, okay? They are not soluble in water. Again, going back to the beginning of the video, we're taking insoluble substances and we're making them soluble. And so fat goes on a much longer digestive journey because it's not soluble from the very beginning. It's insoluble in water in particular. And so we've got to add a lot of substances to it. And finally, right at the very end, when we get to the small intestine, we can finally digest fats and then, of course, absorb. Now, as always, I like to finish off my videos with a terminology recap. You can use these for flashcards and make studying super easy. And these are just some of the words that I've used throughout the video that you should know and be able to use in an exam. First of all, in the very beginning, I spoke about the ability to break down foods and chew them, and we actually call that mastication. It means to chew, and it makes food smaller via mechanical digestion. We spoke about the enzyme amylase, which is the enzyme found in our salivary glands. It's where chemical digestion begins for carbohydrates in the mouth. We spoke about peristalsis, which is the contractual waves of the esophagus as it pushes food down. And we spoke about metabolic reactions being the way in which enzymes do their job. Now, enzymes can be catabolic or anabolic, which means they can build up or break down substances depending on how much you need of that thing. And speaking of enzymes, we spoke about their active sites, which is the area that links in with the substance you're trying to break down or the substrate. And that specific substrate is specific to the enzyme. Now, if you change the active site, you denature it, which means that you cannot use it anymore. And this is very relevant, remember, to the digestive system because we often denature with pH. And so if it's in the wrong pH, the enzyme won't work anymore. It'll be denatured. And last but not least, we spoke about the words insoluble and soluble, which, if you remember, the whole point of this whole journey is to create small particles to increase the surface area so that absorption can take place. Now, if the substance doesn't get small enough, it won't be soluble, it won't move into water, it won't move into the bloodstream. And so if something is insoluble, it's not absorbable and it won't be able to be used by the body. So the point of the digestive system is to make food smaller, increasing the surface area so that it can become soluble and be absorbed into the bloodstream. Now, if you like this video, don't forget to give it a thumbs up, and I will see you all again soon. Bye.